Ever think about those diseases that sort of hide in plain sight? Or maybe have this surprising double life, harmless 1 minute, then bam, real trouble. Well, today we're tackling exactly that kind of puzzle, Glässer's disease in pigs. We've sifted through a ton of material. You shared really deep analyses, historical stuff, and wow, there's some fascinating insight packed in there. So our mission for this deep dive, let's try and cut through the scientific jargon, the complexity. We want to pull out the really crucial bits about this disease. It affects pigs worldwide. But it's not just what it is, it's why it keeps being such a persistent, sometimes unexpected headache in modern farming.
And, while Glässer's disease might sound really specific, maybe even a bit niche, its story is, well, it's a perfect example of how animal health, farm economics, and even some fundamental biology all connect. As we dig in, I think you'll have quite a few aha moments about disease control in general, things that apply way beyond just pigs.
Okay, let's start right at the beginning then. What exactly are we talking about with Glässer's disease?
Fundamentally, it's a bacterial infection that spreads throughout the pig's body. Usually, we recognize it by this classic trio of problems. You get fibrinous polycercitis. It's basically inflammation, sticky inflammation, on the linings of body cavities, like the chest, the abdomen, around the heart.
Right. And then polyarthritis inflammation in multiple joints, making them swollen and painful.
And the third one is meningitis. Inflammation around the brain and spinal cord. Not good.
Not good at all. But what's really striking is how much the actual symptoms can vary from pig to pig or farm to farm. Sometimes, yeah, it looks mainly like a lung problem, maybe severe bronchial pneumonia. Other times, though, it's incredibly fast peracute septicemia. That means the bacteria just overwhelm the bloodstream, and pigs can literally be fine 1 minute and found dead the next with almost no warning signs.
Wow.
Yeah. It really shows you how systemic it can be, how it can hit the whole body. That variability must make it tricky to diagnose sometimes. Now, it's interesting. The disease itself has been known for a while, named after Dr. Carl Glässer back in 1910. But the bug itself, the bacterium, it used to be called Haemophilus parasuis. That name changed, didn't it?
It did. Big change, actually. With better genetic tools, scientists realized it didn't quite fit in the Haemophilus group, so it got reclassified into a whole new genus. Now it's officially Glaesserella parasuis, or GRAPS for short.
Okay, Glaesserella. So not just a name tweak then.
Oh, definitely not. This reclassification, it came from a much deeper understanding of the genetics, the whole family tree of these bacteria. And that has real practical consequences. It means researchers can now develop better diagnostics, better vaccines, targeting more fundamental sort of core features of the bacteria rather than just the surface bits that can change a lot.
Like getting a better blueprint.
Exactly. A much clearer picture of what we're actually dealing with.
So this bug, Glaesserella parasuis, what does it mean for pig farming globally? Is it widespread?
Oh, incredibly widespread. Ubiquitous is the word often used. You find it basically in every major pork-producing country around the world. And what's really kind of amazing or frustrating, depending on your perspective, is that it sticks around even in really modern, high-health status herds.
Yeah.
Places with top-notch biosecurity. It's just there.
Always lurking.
Pretty much. Which leads to the big question, right? What's the actual cost?
What's the damage?
It's substantial. You get high morbidity, lots of sick pigs, and sometimes really high mortality death rates. In herds that haven't seen it before, naive herds, mortality can sometimes shoot over 70%.
70%?
Yeah. And even the ones that survive, they often don't grow well. They don't convert feed efficiently. Then you've got the direct costs, antibiotics, labor to treat them, and a really big hidden cost comes later. Chronic cases often develop these fibrous adhesions like internal scar tissue in the chest and abdomen, and that can lead into parts of the carcass or even the whole thing being condemned at the slaughterhouse. That's a direct hit to the farmer's income.
Is there a figure on that, like per pig?
Yeah. There was one analysis, an industry estimate, that put the extra cost at around $8.60 for each pig affected by Glässer's.
Wow. Multiply that across a large farm. That adds up incredibly fast.
It's a huge economic drain, no question.
Okay, but here's where it gets really interesting, almost weird. This Glaesserella parasuis. It's not always a villain, is it?
No, not at all. And that's the paradox. It's not what we call an obligate pathogen, meaning it doesn't need to cause disease to survive. In fact, it's a totally normal resident, a commensal organism found in the upper respiratory tract, the nose, the tonsils of perfectly healthy pigs.
But he just lives there.
Yeah, piglets usually pick it up from their mothers within the first few days of life, and for many pigs, it just stays there harmlessly.
So how does it flip? How does it go from being like a quiet roommate to causing all this chaos?
That's the million-dollar question. It's not random. That switch, that transition from harmless colonizer to invasive pathogen, it's almost always kicked off by specific trigger factors, things that disrupt the balance,
Yeah.
the sort of truce between the pig's immune system and the bacteria.
Okay, so if it's everywhere anyway, and it's usually harmless, what does that mean for trying to control the disease? You can't just get rid of the bacteria, can you?
Exactly. Eradication just isn't realistic. It's too widespread, too common even in healthy animals. So the whole strategy shifts. You stop thinking about eliminating the bacteria itself. Instead, you focus on managing the disease by managing the factors that allow it to cause trouble. That means focusing on the pig's immunity, reducing stress, and controlling other infections that might give G. parasuis an opening. It's about managing the expression of the disease, not the presence of the bug.
Got it. Okay, let's zoom in on the bug itself then. Glaesserella parasuis. What's it like microbiologically?
Well, it's a small bacterium. It's gram-negative, which tells us about its cell wall structure. It can be pleomorphic, meaning it can look different shapes under the microscope, and it's quite picky, fastidious about how it grows in the lab. It needs specific nutrients, particularly something called V factor, or NAD.
NAD?
Nicotinamide adenine dinucleotide. It's a coenzyme. Because it needs this, labs often have to use special enriched media, like chocolate agar, or they do this trick where they streak another bacterium like Staph aureus on the plate next to it. The staph acts like a nurse, providing the V factor.
Okay, picky little thing. Now, you mentioned diversity earlier. That's been a real challenge, hasn't it, with serovars?
Oh, absolutely. Historically, researchers tried to classify strains based on the sugars on their surface, their capsular polysaccharides. They identified at least 15 different types called serovars.
Right.
But that system had problems. Big problems. For one, a lot of strains they found in the field, sometimes up to 40%, just couldn't be typed with the standard test. They were non-typable, or NT.
That's not helpful.
Not at all. And even worse, they found that how dangerous a strain was, its virulence, wasn't reliably linked to its serovar. Like serovar 7 was originally thought to be harmless, a virulent. But then they started finding serovar 7 causing really severe Glässer's outbreaks. So the whole system kind of fell apart in terms of predicting risk.
So what replaced it? How do scientists tell strains apart now?
Well, the field moved towards molecular methods, looking directly at the DNA. Techniques like ERIC PCR, which is like a DNA fingerprinting method, and now, increasingly, whole genome sequencing, WGS, where you map out the entire genetic code. These give much, much better discrimination. And a really useful development has been multiplex PCR assays, MPCR.
Multiplex, meaning it looks for several things at once.
Exactly. These MPCR tests can identify the bacteria, figure out its serotype directly from the DNA, which cuts down on those non-typable results, and sometimes even detect key genes linked to virulence, all in one go.
Okay, that sounds much more powerful. So speaking of virulence, how does this bacteria actually do damage? What's its strategy?
It's a multi-step invasion, really. It starts in the upper respiratory tract, where it normally lives. Virulent strains have ways to get past the lining, the epithelium. They can actually mess with the connections between the cells, the tight junctions, making that barrier kind of leaky.
Creating an opening.
Precisely. This lets the bacteria slip through into the deeper tissues and ultimately get into the bloodstream.
And once it's in the blood.
Then it has tricks to evade the pig's immune system. It can resist being eaten by immune cells like macrophages, and it seems to delay the pig making antibodies against it.
Sneaky.
Very. And then the really severe inflammation, you see the fever, all that fibrin being laid down, a lot of that is actually the pig's own immune system reacting, maybe overreacting, to components of the bacteria, especially its lipopolysaccharide, or LPS. That's an endotoxin.
So the body's response causes a lot of the damage.
The huge amount of it. It can even lead to widespread blood clotting problems, DIC.
So are there specific genes scientists have found that make a strain more dangerous? These virulence factors?
Yes, definitely. One group that's been studied a lot are the VTA genes. They code for proteins on the surface that help the bacteria stick to things, and they found that certain types or groups of these VTA genes, especially one called group 1, are strongly linked with the really nasty invasive strains.
So you can test for those genes.
Yep. They're now valuable markers in those diagnostic PCR tests we talked about. Helps tell the bad actors from the relatively harmless ones.
Okay, let's shift to how it spreads. Epidemiology, where does it start?
The main source, the reservoir, is the sow, the mother pig. Piglets aren't born with it, but they pick it up very early, usually within days of birth.
Wow.
Mostly just nose-to-nose contact with the sow. It colonizes their upper respiratory tract.
Okay, so they get it really early. But you said the disease outbreaks often happen later, like weeks later. Why the delay?
Yes. That brings us to this critical concept, the immunity gap.
Immunity gap.
Yeah. See, piglets get their first dose of protection from their mom's colostrum, that first milk. It's packed with maternal antibodies.
Right, passive immunity.
Exactly. But that passive immunity doesn't last forever. The levels of those maternal antibodies gradually decrease over the first few weeks of life. This creates a window, typically between about 3 to 5 weeks old, maybe stretching up to 8 or 12 weeks, where the maternal protection is fading, but the piglet's own active immune system isn't fully ramped up yet.
A gap in protection.
Precisely.
Right.
And that time frame, that's exactly when we see most clinical Glässer's disease outbreaks happen.
That makes sense, but I remember reading something almost counterintuitive about early exposure being maybe good.
Yes, the natural vaccination paradox. It's fascinating. Think about it. If a piglet gets colonized by a potentially virulent strain, while it's still got high levels of those maternal antibodies protecting it, it might actually develop its own strong active immunity to that strain without ever getting sick. The maternal antibodies prevent disease, but the exposure still trains the immune system.
So preventing that early exposure altogether might actually be bad.
It could be counterproductive, yeah. Piglets raised in super clean environments, or maybe giving antibiotics early that wipe out the normal bacteria, they might miss that window for natural immunization. Then, when their maternal antibodies finally disappear after weaning, and they encounter the bacteria, they're completely naive and much more likely to get severe disease.
Wow. So a little bit of exposure under protection is key.
It seems that way. It suggests maybe we shouldn't aim for zero exposure, but controlled early exposure.
Okay, so the bacteria present, we have this immunity gap. What are the sparks that actually light the fire, the triggers?
The number one trigger, hands down, is stress.
Stress.
And the post-weaning period, it's probably the single most stressful time in a commercial pig's life. And it lines up perfectly with that immunity gap.
But what kind of stress are we talking about?
It's everything hitting them at once. Psychological stress, being taken from their mother. Physiological stress, a sudden drastic change in diet from milk to solid feed. Environmental stress, moving to a new building, maybe temperature swings. Social stress, being mixed with unfamiliar pigs, establishing new hierarchies.
That's a lot.
It's a massive amount, and each one of those stressors suppresses the immune system, making the pig more vulnerable.
And this often happens alongside other diseases too, right? It's part of a bigger picture.
Absolutely. Glässer's is often described as a classic secondary bacterial infection. It's a major player in what vets call the porcine respiratory disease complex, or PRDC. This means it often follows an initial infection by a primary virus, something that weakens the pig first.
Like what viruses?
The big ones are PRS virus, porcine reproductive and respiratory syndrome, and influenza, a virus in swine, IAVS. These viruses don't just cause general immunosuppression, they also directly damage the lining of the respiratory tract.
Opening the door again?
Exactly. They physically damage the barrier, creating that pathway for G. Parasuis to invade from the airways into the rest of the body.
So controlling Glässer's really means controlling those viruses too.
You absolutely cannot separate them. Effective control of Glässer's is fundamentally linked to having good control over PRSV and influenza.
Okay, so let's picture the pigs. What does Glässer's actually look like on the farm? Which pigs get it most?
As we said, the prime time is in that nursery or early grower phase, usually pigs between 4 and 8 weeks old, right in that immunity gap window. You can sometimes see it earlier in suckling piglets, especially if they came from first-time mothers, gilts, whose colostrum might not have been as rich in antibodies. But mostly, it's those post-weaning pigs.
And the signs vary a lot, you mentioned.
Hugely. We often break it down into forms. There's the paracute form, sudden death. Pigs might look okay 1 hour and be dead the next. That's usually overwhelming septicemia. Then the acute form, the sort of classic Glässer's. This comes on fast, high fever, like really high, up to maybe 106, 107 Fahrenheit. They're severely depressed, off feed, don't want to move. You'll often see them breathing hard, maybe thumping their sides because of the inflammation around the lungs and heart.
The polyserositis.
Exactly. And the polyarthritis makes their joints really swollen and painful, so they're lame, reluctant to stand. And if there's meningitis, you get neurological signs. Muscle tremors, staggering, sometimes they sit like a dog or paddle their legs lying down, arch their backs, convulsions even. In the final stages, they might look bluish cyanotic.
And if they survive that acute phase,
They can go into the chronic form. These are the poor doers. They're stunted, thin, maybe look pale, rough hair coat. This is because those fibrinous adhesions we talked about become permanent, organized, fibrous tissue, like internal scars, restricting organ movement and growth. Chronic arthritis keeps them lame.
the tombstone lesions they find at slaughter.
That's often what those chronic adhesions are called,
Yeah.
because they can really impact the carcass value.
Okay, and when you do a postmortem, a necropsy, do the findings match up clearly with these stages?
Oh, definitely. In those acute cases, the absolute hallmark is that severe, widespread fibrinous polyserositis. You open the pig up, and there's this thick, yellowish or grayish, sticky fibrin coating the surfaces of the lungs, heart, liver, spleen, intestines, the joint linings.
Yeah.
It's everywhere.
A mess.
A real mess. In the chronic cases, that fibrin has organized into firm, white, fibrous adhesions. Sometimes the lungs are basically stuck to the chest wall. You can see why those pigs don't thrive.
Right. So given all this variation and the fact that other things might look similar, how do you nail the diagnosis? It sounds challenging.
It is challenging, and that's a critical point. The clinical signs and even the lesions at necropsy, they are not unique to Glässer's. They're not pathognomonic.
Meaning other diseases can look just like it.
Exactly. Several other common bacteria can cause a very similar picture of polyserositis, especially in pigs that age.
And if you guess wrong,
You waste time and money on treatments that won't work, and the pigs continue to suffer or die. So, a proper diagnosis is key.
so what's the process?
It's a combination approach. You start with the clinical signs, the history, what age pigs, any recent stresses like weaning. Then the postmortem findings, seeing that fibrinous polyserositis is a really strong clue, probably the most important presumptive finding.
Presumptive, okay.
And this is non-negotiable. You absolutely need laboratory confirmation to be sure.
And where do you sample from? Since it lives in the nose normally.
Excellent point. You must sample from systemic sites where the bacteria shouldn't normally be, like fluid from the chest or abdomen, swabs from the surface of the heart or liver showing fibrin, affected joints, or even brain tissue if there are neurological signs. Sampling just the nose or tonsils is useless for diagnosing disease because you'll likely find it there anyway, even in healthy pigs.
Got it. Systemic sites only. And what do the labs do? How have the tests evolved?
Well, the old gold standard was bacterial culture,
Right.
trying to grow the organism. The big advantage is you get a live isolate, which you can then test for antibiotic susceptibility and even use to make a custom vaccine for the farm. But, as we said, G. Parasuis is fragile and picky. It can die during transport to the lab, or maybe antibiotics were already given, preventing growth. So false negatives are common. You need really fresh samples, handled carefully.
Okay. So culture is useful but tricky. What else?
PCR polymerase chain reaction was a big step forward. It detects the bacteria's DNA. So even if the bacteria are dead from transport or antibiotics, PCR can still find them.
That sounds better.
It is in many ways, but again, context is key. A positive PCR from, say, lung tissue with pneumonia is significant. A positive PCR from a nasal swab, less so, could just be a colonizer. So PCR is best used on samples from those sterile systemic sites. And now we have the even more advanced molecular diagnostics, like those multiplex PCR assays. They give you the most comprehensive picture. Is it G. Parasuis? What serotype is it? Does it have known virulence genes like VTA? That really helps distinguish the troublemakers.
Okay, comprehensive picture. Now, about that polyserositis conundrum, what are the main diseases that look like Glässer's? The key differentials.
A big one is Streptococcus suis, another common bacterial infection in pigs. It can cause similar meningitis, arthritis, sudden death.
How might you tell them apart?
Sometimes strep suis causes more pus, a purulent exudate rather than just fibrin. Maybe more obvious heart valve infections, endocarditis, and often the meningitis signs might be even more severe, but it can look very, very similar.
Okay, what else?
The other really critical one to consider, and it's being recognized more and more, is Mycoplasma hyogenes.
Mycoplasma.
Yes, this organism can cause a clinical picture, polyserositis, arthritis in nursery pigs that is virtually identical to Glässer's disease.
Identical, wow. Why is that one so critical to differentiate?
Because of treatment. Mycoplasmas are fundamentally different from bacteria like G. parasuis. They don't have a cell wall.
Okay.
So that means they are naturally, intrinsically resistant to all beta-lactam antibiotics. That whole class includes things like penicillin, ampicillin, ceftiofur, very common drugs used to treat suspected Glässer's disease.
Ah. So if you think it's Glässer's and you treat with penicillin, but it's actually mycoplasma.
The treatment will do absolutely nothing. Zero effect. You're just delaying proper treatment, letting the disease get worse, wasting money. That's a huge aha moment for vets in the field. Distinguishing those two is paramount.
That's a massive implication. Any others, briefly?
You might also consider Actinobacillus suis, sometimes other bacteria, but strep suis and especially Mycoplasma hierarchines are the top mimics you need to rule out.
Okay, let's talk treatment then. Assuming you do have Glässer's confirmed, in an acute outbreak, you need to act fast, right?
Absolutely. Prompt treatment with injectable antibiotics is critical to try and save pigs and reduce the severity. The disease can progress really rapidly.
And historically, were antibiotics pretty effective?
Yeah, for a long time, G. parasuis was generally considered susceptible to a pretty wide range of common swine antibiotics: penicillins, cephalosporins, tetracyclines, macrolides.
But that's changed.
That has changed dramatically. That historical reliance, combined with maybe less than optimal usage sometimes, has led exactly where you'd expect, to the massive emergence and spread of antimicrobial resistance, AMR.
So you can't just assume the usual drugs will work anymore.
Definitely not. Assuming susceptibility is now a dangerous gamble. AMR in G. parasuis is a major global challenge for controlling this disease effectively.
How bad is it? We have numbers.
We do, and they're pretty sobering. Surveillance studies consistently show high resistance rates. For instance, there was a study from Spain published just recently in 2024. They found over 63% of the G. parasuis isolates they tested were multi-drug resistant, MDR.
Meaning resistant to multiple different types of antibiotics.
Exactly, resistant to 3 or more different classes. And in that same study, resistance to macrolides drugs like Tylosin and tilmacosin was exceptionally high. Like over 93% resistant to Tylosin, nearly 98% to clindamycin.
Wow. Almost completely resistant to some classes. How does that happen? How do they become resistant?
We understand the genetics much better now, thanks to whole genome sequencing. Often, it involves the bacteria acquiring specific resistance genes. For tetracycline resistance, it might be genes like TET-B or TET-H. For beta-lactams like penicillin, maybe BLARO-B1.
They pick these genes up.
Yeah, often from other bacteria. And this is where it gets really concerning. Some key resistance genes, like ERM-T, which confers macrolide resistance, are frequently found on what we call mobile genetic elements. Think of them as little packets of DNA, like ICEs, integrative and conjugative elements that can actually jump between bacteria. Literally transfer from one bacterium to another, even to different species living in the pig's gut or respiratory tract. This allows resistance to spread incredibly rapidly through the bacterial population.
That sounds bad.
It's a major driver of AMR spread. Other resistance, like to fluoroquinolones, might arise from small mutations in the bacteria's own genes.
So with resistance being so common and spreading so easily, what's the takeaway for pig producers and vets?
It fundamentally changes the approach. The single most critical implication is that you absolutely need to do antimicrobial susceptibility testing, AST.
Testing the specific bacteria from that farm.
Yes. Before you rely on an antibiotic for treatment or control, you need to isolate the G. parasuis strain causing the problem on that specific farm and test it in the lab to see which antibiotics it's actually still susceptible to.
Creating a farm-specific antibiogram.
Exactly. That's the only reliable way to choose a drug that has a decent chance of working. Just guessing based on historical use is often futile now.
Okay, but is there another layer to this? I read something about resistance being linked to virulence, making the problem even worse.
Yes. That is a deeply troubling finding from some recent research, the potential for co-selection.
Co-select.
It means that sometimes the genetic elements that carry the resistance genes might also carry genes that make the bacteria more virulent, more pathogenic. For example, one study found a strong statistical link between a particular virulence gene and resistance to tetracycline. The genes might be physically located close together on the bacteria's DNA or on those mobile elements.
So if you use tetracycline,
you might not only be selecting for bacteria resistant to tetracycline, but you could also be inadvertently selecting for bacteria that are genetically programmed to be more harmful, more invasive.
oh wow, that adds a whole new level of complexity, doesn't it?
It really does. It makes responsible antibiotic use based on diagnostics and susceptibility testing even more critical than ever. Our choices have potentially bigger consequences than we thought.
Okay, so wrapping all this together, the bacteria are everywhere, there's the immunity gap, stress is a huge trigger, other diseases play a role, and now antibiotics are becoming less reliable due to resistance. How on earth do you effectively control Glesser's disease? Sounds like there's no silver bullet.
There absolutely isn't. Anyone looking for one simple fix will be disappointed. Effective control demands a multi-pronged, integrated strategy. You have to attack it from several angles at once.
What's the foundation? Where do you start?
The absolute cornerstone is solid farm management and biosecurity.
Okay, break that down. What does good management look like for Glassers?
A huge part is stress reduction, especially around that critical weaning period. This means practical things. Making sure building temperatures are stable and comfortable, ventilation is good to maintain air quality, avoiding overcrowding pigs in pens. It also means focusing on low-stress weaning techniques, handling pigs gently, maybe even using nutritional strategies like certain amino acids, probiotics, prebiotics to support gut health and reduce physiological stress.
Makes sense. What about how pigs are grouped and moved?
This is where all in all out AIAO production comes in. It's arguably one of the most powerful management tools we have for breaking disease transmission cycles, not just for Glassers, but for many diseases.
All in, all out.
You manage pigs in distinct groups, usually matched by age. An entire group moves into a cleaned, disinfected barn or room together. They stay together, ideally without mixing. Then the entire group moves out together. The facility is then completely emptied, washed, disinfected, and allowed to dry, given downtime, before the next group enters.
Breaking the cycle.
Exactly. It prevents pathogens from building up and passing from older, potentially shedding pigs to younger, susceptible ones. But, and this is crucial, the system only works if it's strictly followed. If you start mixing pigs between groups or holding back tail enders to mix with the next batch, you completely undermine the benefits.
Got it. All in, all out, strictly. What about keeping new problems out?
That's biosecurity, rigorous protocols to prevent new, perhaps more virulent strains of G. Parasuis from getting onto the farm in the first place. And just as importantly, keeping out those primary viral triggers we talked about, like PRRSV and influenza. Good biosecurity protects against multiple threats.
Okay, management and biosecurity are the base. What about specific interventions like vaccines?
Vaccination is definitely a critical tool, but it's also complex, largely because of all that strain diversity we discussed earlier. There isn't one perfect vaccine that covers everything.
What are the main types available?
Broadly, there are three categories. First, you have commercial bacterians. These are traditional vaccines made from inactivated, killed G. Parasuis bacteria.
Pros and cons.
They're generally very safe. The downside is that the protection they offer is often specific to the serovar included in the vaccine. They might not protect well against different strains circulating on the farm, so cross-protection can be variable or limited.
Okay, type 2.
Commercial virulent live vaccines. There's one well-known example, Parasuis. These use a live strain of G. Parasuis that has been attenuated, meaning it doesn't cause disease, but can still replicate a bit in the pig and stimulate immunity.
Advantages.
They tend to induce a more robust and potentially broader immune response, possibly including better mucosal immunity in the respiratory tract and cell-mediated immunity, which bacterians don't do as well. They're often given as a single dose.
Any catches?
The big one is you absolutely cannot use them at the same time as antibiotics that would be effective against G. Parasuis, because the antibiotics would simply kill the live vaccine organism before it could stimulate immunity. Timing is critical.
Right. And the third type?
Autogenous vaccines. These are custom-made bacterians.
Custom.
Yeah. You isolate the specific pathogenic strain of G. Parasuis that is actually causing disease on that particular farm. Then a licensed facility uses that isolate to create a killed vaccine specifically for that farm.
Sounds very targeted.
It is. That's the main advantage. It targets the exact strain causing the problem. The limitations are you first have to successfully isolate the bug, which isn't always easy. Then it takes time, usually 6 to 12 weeks, to manufacture the vaccine. And like commercial bacterians, it might offer little to no cross protection if a new strain gets introduced to the farm later.
So different tools for different situations. How are they used? Vaccinate the piglets or the mothers?
Both strategies are used. You can vaccinate the piglets directly, usually starting after weaning, often with a 2-dose schedule. The goal is to stimulate their own active immunity as their maternal antibody levels are dropping. Alternatively, or sometimes in combination, you can vaccinate the sows and gilts before they farrow, before they give birth. The aim here is to boost the level of antibodies in their colostrum.
So the piglets get higher passive protection.
Exactly. Higher maternal antibodies to help protect them through those first critical, vulnerable weeks of life, bridging that immunity gap.
With all this complexity around strains, what's the ultimate goal for vaccine research? The Holy Grail.
The holy grail would be to identify parts of the G. Parasuis bacterium-specific proteins or antigens that are essential for its survival or virulence, AND, are highly conserved, meaning they don't vary much between different strains and serobars. If researchers could find such conserved protective antigens, they could potentially develop a universal vaccine that offers broad cross-protection against most, if not all, virulent strains. That would be a game changer, overcoming the current limitations of serovar-specific immunity.
A universal vaccine, that would be huge. Okay, so we have management, biosecurity, vaccination, anything else in the integrated approach?
Yes, and it circles back to something we mentioned earlier, the absolute necessity of integrated health programs. You simply cannot expect to control Glässer's disease effectively in isolation if you have uncontrolled circulation of those primary immunosuppressive viruses, especially PRRSV and swine influenza.
They just keep opening the door.
Constantly. So any successful Glässer's control program must include robust strategies for preventing and controlling those key viral pathogens simultaneously. It's all interconnected.
Okay, so bringing it all together then, Glässer's disease. It really sounds like a disease that's almost been shaped by modern intensive pig farming, doesn't it?
I think that's a very accurate way to put it. It's this incredibly complex dance between a bacterium that's normally harmless and ubiquitous, the natural decline of maternal immunity in young animals, and the immense pressures, physiological, social, environmental, that we place on them, especially around weaning.
A biological puzzle created by the system itself, in a way.
In many ways, yes. And the path forward for managing it really requires a shift in thinking. We have to move away from primarily relying on reactive treatment with antibiotics after the problem starts.
Especially with resistance climbing.
Exactly. The escalating AMR crisis makes that old approach increasingly unsustainable, sometimes simply ineffective. True success is going to come from a more holistic view. Understanding all these interacting factors, the bug, the pig, the environment, the other pathogens, and committing to proactive preventative strategies, things based on science, focusing on robust management, smart vaccination, and overall animal health and welfare. The goal is to reduce the need for therapeutic intervention in the first place.
Prevention is better than cure, especially when the cures are failing.
Precisely.
So a final thought for everyone listening. As you go about your day, maybe think about this delicate balance. A host, a microbe that's always around, and environmental factors or stresses. Does that pattern ring any bells in other areas, maybe in human health or other animal systems or even non-biological systems? Where else might stress be cracking open the door for opportunistic problems, turning something that was manageable into a real issue? It's something to ponder. Keep looking for those connections, those aha moments in whatever you're learning about.